I hope to bridge the division in some senses between biology and materials. So I'm learning a lot by meeting you all here. Okay, so I'm going to talk about the natural material, the arrangement of the polysaccharides in the wood itself. I come from the biological perspective, as I say, and so I'm interested in the cell walls themselves in the plants from the perspective of trying to understand how plants grow, how the cells expand, and the role of the polysaccharides in that process. And then in the secondary cell wall, what is the role of these different polysaccharides in making the strong materials? And of course, I have an eye on a wide range of different types of applications, such as the ones we're talking about today, but also interest in food quality and polysaccharides as animal feed, and even in dietary fibre. And I'm a co-founder of a food ingredient company, for example, using xylans and other polysaccharides. Okay, so I'd like to acknowledge the funders at the start, and also the people who've been involved in the work that I'm going to talk about. In particular, Yoshi and Lee are taking forward the work on Manan that I'll mention briefly, and students Jan, Nick, and Oliver were very productive in my lab in studying the molecular architecture of cell walls. And Tom was involved as well in the early solid-state NMR work. Now, I work with a lot of collaborators. I'd particularly like to mention Ray in the Sainsbury lab in Cambridge, who does electron microscopy, and the National Solid-State NMR facility in Warwick, and particularly a wonderful postdoc, Rosalie Cresswell, who works with my father, Ray Dupree, in the solid-state NMR. So it's a nice family collaboration. Okay, so what are these big questions? So we know lots of things about why plant cell walls are so marvelous. They have these strong, rigid cellulose fibrils within the walls, and it's embedded in other polymers. And we know that xylan and hemicelluloses somehow interact with these cellulose surfaces, and lignin surrounds it, and this excludes water and enzymes, etc., and builds up the strength of the material. But what we don't understand well enough, I think, is how the wall is assembled in the biological process, what leads to these precise arrangements in the cell wall, and also what are those precise arrangements in the cell wall, and are they really important for the properties of the cell wall and the woody materials. So that's what I've been approaching over the last few years, to try to understand how plants build up the material and what that structure is, and then what happens if you disrupt that process. So thinking at the larger scale, first of all, large in the sense of multiple nanometer scale, first of all, we were interested in how the polymers might bundle together in the cell wall, and there are reports of aggregations, bundling of cellulose fibrils, and even formation of so-called macrofibrils. And this was done by Lloyd Donaldson in New Zealand in dry material. So we wanted to look in wet material to see if any of that is a function of drying of the cell walls. So we used this cryo-scanning electron microscope in the Sainsbury lab in Cambridge, where we take fresh Arabidopsis stems and snap freeze them, and then fragment them, just snap them, and look at the surfaces of the cell walls. And here in our model plant, Arabidopsis, you can see vessels, and they're surrounded by fiber cells. And if we zoom into the cell wall, the secondary cell wall around the vessels, eventually you can see lots of regular points or slight long, thin structures, which we considered are these bundles of cellulose fibrils together with other components in the cell wall. And it is somewhat reminiscent of cellulosic materials extracted that you're more familiar with, perhaps. So has this got anything to do with cellulose and the other components in the wall? So what we can do is use molecular genetics, so genetic manipulation of plants, to add and remove components and see what happens. So the first question was, what happens if we remove all cellulose from secondary cell walls? And here on the right, we have so-called IRX3 mutant plants, which are unable to make any cellulose fibrils in the cell wall. And on the left here is the wild type, the normal cell walls. And here you can see these structures. Lots of points where we've broken off these bundles, which are about 20 nanometers across. And in the cellulose deficient mutant, the walls are amazingly smooth, so there's nothing there. So not really surprising, I suppose. These things really are bundles of cellulose, and their cellulose is necessary to make them. But that proves the principle. So what about the other components? Well, in my lab and many labs around the world over the last 20 years, there's been tremendous progress made in understanding how plants make the hemicelluloses. It happens in the Golgi apparatus. There is an array of membrane-bound enzymes that polymerize the xylan backbone and add the sugars, such as glucuronic acid and acetate, to this backbone to keep it soluble, as we've heard. The mannan is also made here in the Golgi. So we now know how these polymers are made. We can manipulate the backbone synthesis, and we can manipulate the acetylation and the sugar substitutions. And we can see, what does this do to the properties of these hemicelluloses, and what does it do to the assembly of the cell wall? So looking now back at these bundles of macrofibrils to see how they change when we change polymer hemicellulose synthesis. So in a normal plant, they are about 18 nanometers diameter with a wide range measured, but 18 on average. In the case where we reduce the amount of xylan or make the xylan shorter, we do see an effect on the average width of these bundles. Similarly, if we change the amount of lignin in the plants, they're also slightly smaller, but they're still seen. When we remove glucomannan, there was no effect. So we get here then an idea, an indication that xylan and lignin are necessary for the correct assembly of these macrofibrils. They can still form, but they're not as large. And that raises, though, many more questions. So is it simply coating on the outside of these fibrils, macrofibrils, or are they somehow inside the macrofibrils? Why is the diameter smaller? We've been doing more specific manipulations of the polymers to try to get at this point. And the first manipulation I'd like to emphasize is the question of adding glucuronic acid or glucuronidation to the xylan. In all plants, all vascular plants, the xylan has glucuronic acid. This is true whether you're a moss, which is not a vascular plant, or going through to gymnosperms, conifers, or hardwoods such as birch. They all have glucuronic acid on them. Why might this be important? So we discovered the enzymes that add these glucuronic acids, and we removed them in Arabidopsis. Surprisingly, the plants grow fine. This is our little weed, and you can see the stems look entirely normal. But the xylan there has no glucuronic acid. So after many years of trying to find out what happens in this case with no glucuronic acid, we do find actually there are some very interesting subtle differences to the cell wall. And one of the most remarkable is its resistance or lack of resistance to enzymatic digestion. So in this case, we take the secondary cell walls, the stems, the straw, if you like. We do some mechanical disruption, but no chemical disruption, and then add enzymes to digest the cellulose and the xylan. On the left here, we see the amount of glucose released from the plants, and on the right here, the amount of xylose. And we tested the effect of reducing acetylation of the xylan by half here, or removing all of the glucuronic acid in this case. And we saw marginal differences in the digestibility of cellulose, but the xylan became remarkably digestible, 7 times as much xylose released. And this is no chemical pretreatment. Let me emphasize this again. This is just mechanical disruption. And so there's something about the glucuronic acid on the xylan that is really important for stopping enzyme digestibility. And our current hypothesis is that this is because of a glucuronic acid lignin linkage, which has been quite widely reported, that the lignin esterifies to the carboxylic acid on the glucuronic acid. And in these plants, there is no glucuronic acid, so the lignin is no longer tethered to the xylan. And further studies of these plants suggest that the wood is more porous. There are larger pores where there's water, and this maybe allows enzymatic access into the material. So xylan is important for interaction with lignin in the cell wall. What about further studies on the xylan and other decorations? So it turns out that different plants have quite diverse sugar substitutions on their xylan. They all have glucuronic acid, as I mentioned, but in the case of dicots, such as birch, we have acetylation and glucuronic acid. Conifers have no acetylation, and arabinose and glucuronic acid. So what we found is that there's a very specific arrangement of these sugar substitutions. It's not only they are quite dispersed, but they are always, or nearly always, on even-spaced sugar residues. What do I mean by that? That means quite rarely you get adjacent substitutions. You do find them, but quite often they're 2 sugars apart, or 4 or 6 apart. And in conifers in particular, the glucuronic acid is nearly always precisely 6 xylose residues apart. This is not going to show. Here we go. No. Here we are. So we always count 6 xylose residues apart. So why have plants evolved to put their sugar decorations in such precise positions? So the idea that we had was that it could be to do with the shape of the xylan in the cell wall. And this makes sense to us if the xylan folds flat like the glucan chains in a cellulose fibril, as shown here on the top. If xylan is flat as a flat ribbon, then glucuronic acids or acetates 2 or 4 units apart will be on one side of that ribbon, whereas if they're odd, they will be on opposite sides. And this could have implications for how the xylan interacts with other molecules in the wall. Now, we also find actually in solution, the xylan is a 3-fold screw we find in solution. So this shape of xylan is not the default state. Okay, so what would this flat form do? So molecular dynamics modeling with Munier-Scaff in Brazil indicated that it is at least feasible that xylan in this 2-fold screw could dock onto the surface of cellulose fibrils into so-called crystal vacancies, if you like, on the surface of cellulose fibrils. These are fibrils that we modeled a few years ago now before we adopted different crystal habits, and they have grooves on their surface. But I think the principle is the same no matter what the habit of the cellulose fibril, in other words, the arrangement of the glucan chains. A xylan that is flat could dock onto the surface and extend the crystal. So I like to think of this, it's not quite crystallization of xylan, but it's related in a way. It's extending the crystal of the cellulose by another layer. And it seems to us that having the even spacing would allow this xylan to dock onto the surface in a way that a random pattern of acetylation or glucuronic acid would not. So what about some evidence for that? So we turn to solid-state NMR to do this. So we grow our plants in a 13 CO2 atmosphere, and we take the fresh stems, no drying of the material at all, no preparation, and those stems are put into the rotor for the solid-state NMR. So we're looking at the arrangements in the natural material. This is an inadequate, so-called inadequate spectrum, CP inadequate, where the main peaks here we see are cellulose, and those are labeled with blue carbon numbers, such as carbon, so it's a bit awkward to use. Here we go, carbon 1 of cellulose and carbon 2 of cellulose here, and we have 2 carbon 4s, which correspond to the surface and interior regions of the cellulose microfibril, for example. Now, what about the xylan? So xylan in solution has well-known carbon shifts, and carbon-3 of xylan is around 78 ppm, and we see this in the cell wall of these plants. But most of the xylan had a different shift of carbon-4, shown here at 82 ppm. And that indicates that the xylan has two conformations in the wet natural material. And what could those two conformations be? Well, a change in carbon-4 shift, and there's also a carbon-1 shift, suggests that it's a change in the glycosidic bond angle around the 1,4 linkage in the backbone of xylan. So this is clear evidence xylan changes its conformation in the cell wall. Now, has this got anything to do with interaction with cellulose? So I told you that we have plants where there is no cellulose in the secondary cell wall. So we compare the spectrum of this cellulose deficient mutant in orange with this wild type plant to see what difference there is. Now, there's a little bit of cellulose left in these plants because we have primary cell walls, but the main difference that we see is a reduction in cellulose and also a change in the xylan shifts. So in the cellulose deficient mutant, we essentially don't have the two-fold screw flattened xylan, and that tells us in the assembly of the cell wall, we need cellulose in order to change the conformation of xylan. And therefore, this is, we think, a xylan-cellulose interaction that's causing this change in conformation. And through space solid-state NMR experiments also indicate the xylan is close to the cellulose. Okay, now what about these substitutions and the patterning? So we have a plant where we've reduced the acetylation, and this leads to a more random patterning of the substitutions, and this plant is called Eskimo. It's cold resistant. Don't ask me about, well, ask me about that later if you like. And this plant, it doesn't grow very well, but we can get it to grow in our growth chambers sufficient for these experiments. And what we find here, so the question we're asking is, what happens if we change the patterning of the xylan? Does this change the assembly of xylan onto the cellulose? So in this case, the orange spectra here are of our changed acetylation plant. And what you can see is that, again, the signals for the two-fold screw xylan are very much reduced in this mutant, and the signals for the three-fold screw xylan are very much more increased. And our recent studies are now looking at the hydration of this, and it seems that it could be that this three-fold screw xylan is more hydrated than in the wild type. So what happens then? If we don't have the correct patterning, the xylan, again, cannot assemble onto the surface of the cellulose. So what we conclude from this work so far then is that the cellulose has quite a large amount of coating with this patterned xylan in hardwoods. Why do I say it's quite a lot? Because if you consider the ratio of xylan to cellulose in the cell wall, and the fact that most of that is two-fold screw xylan, and that requires cellulose, it means probably that much of the surface must be coated in a single layer of this xylan. That is our current hypothesis. And then we also know that the glucuronic acid that's here on the xylan is needed to tether the lignin close to this. So the xylan is an intermediary, if you like. It's a biocompatibilizer, we think, in some sense, between the cellulose, which otherwise would have a hydrophilic face, to making a more hydrophobic face that is chemically linked across to the lignin. Okay, so what about softwoods? So I've talked here about Arabidopsis hardwood, so that's birch. So softwood is quite different, and it has a lot of glucomannan and much less xylan, and there has been some discussion in the literature about whether the xylan has a different role in softwoods. For example, this model from Terashima et al. Suggests that the xylan is between cellulose fibrils, but not close to them, and close to the fibrils is mannan and lignin. So we looked for the presence of xylan and mannan in spruce. So we 13C labeled spruce and carried out this solid-state NMR again. So in black here you see Arabidopsis, and in orange is the spruce wood. And you can see there's a lot of similarities. Okay, it's cellulose, not surprising. One of the big differences is an abundance of glucomannan in the spruce, so we have extra peaks down here for carbon 1 of mannan, mannose, and carbon 2 here, which are different depending on whether or not they're acetylated. The glucomannan in conifer, in softwoods, is partly acetylated. Now, what about the xylan conformation? Well, we can very clearly see this twofold screw xylan in the conifer situation, and very little of the threefold screw xylan. So this tells us that, contrary to some opinions earlier, the xylan really is tightly associated with the surface of the cellulose fibrils in the softwood. And we can do through space experiments to confirm that as well. What about the mannan? Mannan doesn't normally have such a beautiful pattern structure as xylan, but it does seem to change its conformation in order to bind to cellulose in these hydrated natural cell walls. And when we looked at the shifts, the chemical shifts of the mannose residues in the cell wall, particularly carbon 4 of mannose, which again is part of the glycosidic bond, adjacent to the glycosidic bond, this carbon 4 chain is different measured in the cell wall in solid state NMR versus in solution, suggesting perhaps a conformational change, and we'd really like to understand better what that shape of mannan is when it's interacting with cellulose. So we sketched out a crude model of what those macrofibrils might be that I showed you by cryo-SEM at the beginning, considering that they are 18 to 20 nanometers in hardwoods. In softwoods, they're even bigger. They're perhaps 30 to 40 nanometers. This means that they must consist of a large number of individual cellulose microfibrils, which we think are probably 18 chains each. And we know that they are individually coated by some xylan as a twofold screw on some surfaces. And we know that the mannan is also tightly associated through this solid state NMR. And lignin is a little bit more distant. And in this model, we drew the lignin around the edge, but actually it could be interpenetrating quite a lot as well, this macrofibril. So all these components in softwood are necessary probably for assembly of the macrofibril. In Arabidopsis, the mannan is very minor, and we didn't see an importance for it yet at this stage. Okay, so what about water? Because I think this nice meeting is about water, and we recently have been asking questions like what changes in the cell wall when you dry the samples. Everything we've done so far was never dried fresh material. So Rosalie carried out some experiments where some pine, 13C labeled pine, was dried in the oven to complete dryness, and we compared the NMR spectra in the fresh material, the dried material, and then rehydrated material. And this is a through-space experiment, a CP-PDSD through-space experiment where we can resolve individual carbons in the cellulose and the mannan, and these are labeled here. And in blue is the wet material, initial material, and red is the dried, oven-dried material. So to summarize, what we saw is in the dried material, the spectra become broader, perhaps not surprisingly. But also we see that the mannan has a slight change in chemical shifts, particularly in carbon 1 and carbon 6, suggesting a changed conformation. And we can see that here, in this region here. These are cross peaks between carbon 1 and carbon 4 of the non-acetylated mannitol residues, or the acetylated ones in the cell wall. And there's a very subtle shift. So maybe the mannan is somehow being stretched or compressed onto the cellulose surface. Through-space experiments suggest that the whole material becomes a little bit more compact as well. There are stronger cross peaks between the different components. So water is spacing things out. The water is removed, it condenses a little bit. Now, on rehydration, most of these changes were reversible, we found. But the main non-reversible change was that a fraction of the glucomannan became completely mobile and I think therefore solubilized. So the the adhesion of the mannan onto the cellulose is not completely reversible in this drying experiment. When you add the water, some of it solubilizes and washes away. So that's perhaps an indication of one of the changes when wood is completely dried. So this last slide summarizes some of our current thinking then on the changes when we dry and rehydrate pine wood. So this here is the natural state of a single microfibril within that big bundle, with xylan chains here bound to the surface, mannan also bound and lignin a little bit distant, and there's water all around here, the surface. When we dry completely, there's some change in conformations a little bit. Things become closer, the different components become closer together. And when we rehydrate, it goes back to the starting position mostly, but there is some mannan that is no longer associated with the cellulose and becomes soluble. So this leads to quite a lot of ongoing questions, though, because we still don't know how the glucomannan is bound to cellulose. We don't have a good model for that. And that is something that we are going to be working on in a new ERC-funded project in the next 5 years. We're interested still to resolve the question, is xylan really bound to the hydrophilic faces, as I show here in this model? Although we know it's bound, we really actually don't know this mode of binding precisely. And we'd also want to know a little bit more about how the macrofibril is organized, how those microfibrils are held together in that larger assembly. And we'd like to understand better how the different polysaccharides lead to that assembly of these relatively uniform macrofibrils in the wood material. Okay, I'm going to leave those questions hanging there and see if you have any questions. Thank you.